Okay, so let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. And welcome and thank you, SynBYSS people, for joining us the 84th seminar, and I call it Columbia University Day. I am in Singapore at an inconvenient time for this seminar. In other words, very late night at 11 p.m. But I am so excited to meet you after my seminar at Nanyang Technological University today, after 24 hours of air travel, and also to see you all, many of you, at the Metabolic Engineering Conference this Sunday. Along with the conference chair, Matthew Chang, will have the largest number of attendees in the history of this conference series, close to 700 people in the conference venue, with amazing invited speakers, including Professor Sang-Yup Lee, Chris Prader, Jim Liao, Humin Zhao, Greg Stephanopoulos, and Jens Nielsen, Jay Kiesling, Michael Koch, and Carl Harper, among many. So see you soon. So, okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Scott Banta. He doesn't need any introduction. He is the department chair of chemical engineering at Columbia University. He is a great scientist and engineer, and especially a very hard worker. I heard from him that he had multiple part-time jobs during his college years, including working at a bar at night, finishing homework there. He is a role model of many, advising my good colleagues, Ian Wilden and Mark Brenner. So he's not only hard worker, but also good teacher, basically nurturing future leaders. So I'm really appreciate his contribution to the society, scientifically and also educational aspect. So Scott, thanks so much for your great contribution to the field, and especially again for nurturing the future leaders. So virtual podium is all yours now. Thank you so much.
Oh, great. Thank you. Thank you so much, Tae-Suk. I really appreciate the opportunity to talk to everyone today. And I think it's great that you've been doing this. And it's a really nice resource for everyone, and especially the junior faculty and junior colleagues who get an opportunity to showcase and highlight their work. And Jorge, it's nice to meet you. I want to apologize in advance. I'm going to have to jump off after my part. So I'm traveling and actually double booked today. But I will watch the YouTube video later. And but yeah, I was just going to give you a few minutes. I have a couple slides here, just sort of talking about my background a little bit and how I got to where I am, and maybe some advice useful to other people. Can you see that? Yep. Okay. Yeah. So as Tae-Suk said, yeah, I'm a professor and chair of chemical engineering at Columbia University in the city of New York. I'll just sort of walk through a little bit of how I got to where I am. As Tae-Suk said, I kind of had a non-sort of traditional background getting into this business, maybe compared to some others. So when I graduated from high school, I went to college. Wasn't really sure what I wanted to major in. I initially started off at a small teachers college, actually, double majoring in math and physics. Didn't really even know much about engineering. I didn't have any engineering in my family. No one in my family had a higher degree. So it wasn't really something I knew that much about. I was told as a math and physics double major that I would be lined up to be maybe an actuarian, which after I looked up what that meant, realized it wasn't for me. No offense to actually any actuarians in the audience. But so I transferred to UMBC, University of Maryland, Baltimore County, which was near where I grew up. And I went to some advising there, and they told me about engineering, and they told me about chemical engineering, mechanical engineering, all different things. And there was some explanation about how chemical engineering was the hardest major and that they made the most money. So that made sense to me at the time. So I signed on for chemical engineering for no other really great reason, which is, funny to me now. But it was good. Actually, as I moved through the chemical engineering curriculum, I realized that I loved kinetics and I loved biological problems, and I loved, I took a protein engineering class as a senior and realized that's really what I wanted to do. So graduated in '97 from UMBC, go Retrievers. And then I went to Rutgers for graduate school, partially because they had a big NIH training program in biotechnology. And by that point, I had realized I wanted to work in the biotech space. And so I got my master's degree and PhD at Rutgers in 2002, feeling old. And then I signed up for a postdoc at the Center for Engineering and Medicine at Harvard Medical School and Mass General Hospital with Martin Yarmish. So for my graduate work, I started working in the area of protein engineering. My advisor was actually in the molecular biology and biochemistry department. So sort of felt like I got 2 PhD degrees. I satisfied all the requirements for chemical engineering, but I also spent a lot of time learning about, life in a molecular biology and biochemistry department. I was the only engineer in the building. My project there was very much focused on protein engineering. We were interested in engineering cofactor specificity of an enzyme for an industrial process for making vitamin C. So that's the sort of imagery down here, and work that I did as a doctoral student. So after doing a lot of work on one enzyme and one protein, when I was searching out postdoctoral opportunities, I really thought it was a good idea to sort of diversify or broaden my experience. So I sought out a postdoc in metabolic engineering, now sort of looking at whole tissue systems and organs and understanding how metabolites flow through biological systems, as opposed to looking at just a single enzyme. And I thought the sort of dual experience of sort of molecular engineering and then pathway engineering would be good. At the time, we called these things, protein engineering and metabolic engineering. This was before the synthetic biology had been coined. I thought I was well set for a faculty position, so I applied and was accepted in Columbia 2004. And I was so excited. I mean, I really, came from pretty humble background and really thought that, I was really super excited they gave me a chance to be there. I went back and looked at my slides that I put together when I was applying for my job, and I cleverly created something like this as my pitch to a faculty position, that I was going to combine protein engineering and metabolic engineering, and we were going to solve big, important problems, good assistant professor stuff. It's fun that I still have all these PowerPoint slides that I used to do. Color choices are skeptical, but yeah, so it's fun now to look back. I'm coming up on, I just finished my 19th year at Columbia. I've been department chair for the last year. So looking back on now, kind of what I've accomplished in that time. And I think, to me, one of the lessons that I learned looking back at my experience was, I started realizing at the time that, there were certain things that I was good at, but I wanted to sort of diversify that into other areas. And I was pretty fearless about trying to take what we could do, but apply it into new spaces. And so, early on, we did work, with, I don't know if you can see my, make this into the pen. Can you see? Oh,
yep.
So I want the, well, anyway, whoops. All right. Wanted the laser pointer. So what I really wanted to do, that's better, is continue doing protein engineering type work. So we did a lot of classical enzyme engineering for cofactor specificity, substrate specificity, different things like that. But I didn't want to just do that. I thought that as an engineer, and to try and get attention and to try and push the field forward, we really wanted to work on new and interesting types of problems. So we really inspired early on by looking at different things that were happening in sort of the material space, trying to get proteins to assemble into materials. We were also really inspired by a lot of electrochemistry, partially because that's a strength at Columbia, and some of my friends at Columbia were working in that area. And so I tried to start to figure out how can we start moving protein engineering or biological engineering concepts into things that would be of interest in the biochemical, bioelectrochemical space and biosensor space. So we started making, protein biomaterials that were also electrocatalytic, or trying to make, different, biological fuel cell devices using engineered enzymes. These ideas of trying to take what we were good at and sort of push it into new spaces. We started making, biosensor type materials and self-assembling materials that could be used. This is, materials that can degrade organophosphate hydrolase, and turning that into, different technologies, trying to interface, proteins with DNA and using that as a mobilization strategy so that we can control the assembly of proteins on different surfaces. Up here in the corner, trying to figure out how to attach nanomaterials or nanoparticles to enzymes, and then trying to understand what that does from a protein engineering or a protein science point of view, and how that affects catalysis and how that affects kinetics. And so sort of an early theme early on, and still throughout my career is, figuring out what we're good at, and then trying to find all new different ways where we can deploy that, so that we're constantly kind of working on new things, but sticking with the sort of technologies that we know. I'd say sort of right now in my lab, we still have a lot of protein engineering sort of activities sort of across the top. We do a lot of work with this protein here, which undergoes an interesting structural transformation, and different technologies that we can do with that. But even in this example here, we've taken this one peptide, and over the last, say, 15 years, we've engineered it for self-assembly to make biomaterials. We've engineered it for biomolecular recognition, for chromatography separations. We've engineered it to bind and separate rare earth elements. We've done a lot of stuff using it as a switching mechanism in enzyme domains, so we can use this sort of folding mechanism to turn on and off catalysis. Like, we're constantly trying to invent new ways to use these tools that we have and try and deploy them into sort of new spaces. We spent a lot of time thinking about enzyme kinetics. That's maybe my first sort of passion, is thinking about that. And so one of the things we've been doing recently, this is a paper I'm really, really proud of, where we really wanted to try and think about what are the right figures of merit to use to compare more complex enzymes. So a lot of people are familiar that, if you want to make a better enzyme, you should improve Kcat and Km. But if you run through the derivation of that, that really applies mostly to simple enzyme systems, like a simple Michaelis-Menten enzyme. We need to more complicated. We do a lot of work with, multi-substrate dehydrogenases and oxidoreductases. Then this idea of just chasing Kcat over Km is not always appropriate. I think people fail to realize that. And so trying to understand, what's the right sort of engineering approach to use to validate and compare, enzymatic systems. And we're doing some work now with sort of DNA origami and applying that to our enzymes, and thinking about how we can use, mechanobiology and different sort of, nanotechnology approaches to understand how that impacts catalysis. A big unexpected thing that happened in the last 10 years is where I got really involved in also this sort of space of electrofuels and biofuels, trying to think about how to put, renewable electrons into biochemical systems. So sort of the opposite of a fuel cell. In a fuel cell, you're trying to use, you're trying to oxidize a carbohydrate to make electricity. About 10 years ago, 15 years ago, we got involved in the reverse process, which is how can you take renewable electricity and try that to reduce carbon to make fuels and products the other way around. And so these are, this took us down the road of working with some extremophiles, especially cells that are involved in growth on metals. So this sort of protein or biological inorganic materials interface, and how can you think about cells that grow on inorganic materials as a way to give them energy. And that turned into a whole interesting topic of stuff that we're still working through in biomining, and cells that form, structures on and interact with inorganic materials. And 25% of the world's copper is actually produced using bacteria. So trying to understand how they do that and how we can make them better has a huge impact. And recently we've been involved now in rare earth separations using proteins to help bind and separate rare earths, and things that we're interested related to are sort of mining work that sort of originally came from some fuel cell work. So this is a slide I showed all at the beginning of my presentations. Like, this is kind of how I like to see my career go, or research go, where, we do a lot of work at the sort of the atomic level, where we can go in and using protein engineering and change around different side chains, or have some control of different atoms within the molecule. But then we like to, take a look at those mutations, or see what that happens then, more at the molecular level, where we can say, okay, now we have now made a better catalyst, or we understand, some kind of electron transfer mechanism that happens within a molecule. And then we try and see what we can do to move that into sort of bulk properties. Can we make, self-assembling materials, or can we make, systems on electrodes, and try and understand what happens at sort of more of a systems level. And then always try and drive that into some sort of application. Often it's been energy or defense applications, or, energy applications, but other things as well, sensors and things that we do, and some separations technologies. And then if things go really well, you'd like to be able to close the loop and say, okay, understanding, can we relate now these the changes that are made at, changing one side chain over here, can we understand how that affects, performance in a fuel cell or something like that? And if you can close that loop, that's a pretty good place to be. That's a pretty good way to run your career if you can. It doesn't always work out that way, but I always sort of have these sort of design cycles in my head when I'm thinking about the types of projects that we're going to want to work on. I don't want to just make mutations because we can. I don't really want to make devices and not understand how they work. But if you can try and figure out how to really understand your system at the fundamental level, then you can really try and make something better on the back end. And then from there, try and pick and choose applications that are important or impactful or useful. So just sort of summarizing some of what I said, this is now maybe my more, instead of looking at the little Venn diagram I made when I was looking for a job, thinking where I am now, we do a lot of stuff on the front end that sort of falls under the more sort of basic sciences and applied sciences, things that we do. And then we apply that to a whole bunch of different areas and different things over the years. And to me, doing this for 20 years, this is the best part of the job, is that I've been able to work myself into a whole bunch of different applications and get involved in a whole bunch of different things that keeps it all interesting. And so we're not just doing the same thing over and over again. So I've been to copper mines, I've been to, worked on explosive sensing. I've worked on making, high impact materials. We've done some drug delivery and stuff. We're interested in electronic, like all sorts of different things. That's what keeps the job fresh and keeps the motivation high and, gets me excited, which gets the students excited. And, I think that's a big part of it too, is work on things that you're interested in and passionate about. And then it makes everything, easier. So just some closing thoughts. I was out with some people, I'm traveling today, and I was out with some people at dinner last night talking about that I was going to give this talk, and I was running through my list here of things I wanted to say. So, just like I just said, try and work on things that you're passionate about or excited about. Work with people that you like and that you trust. Collaborations are a big part of this job, and when you work with great collaborators, it makes everything better. It's very difficult when you get involved in a relationship with someone that you don't like or don't trust. So try and avoid that. Get out of the office as much as you can. No one knocks on your door to give you big opportunities. You got to go seek them out. And I have lots and lots of stories of being in the right place at the right time leads to some new connection or some new interaction or some new opportunity. And so Tastehook travels more than anyone I know. You got to get out of the house and good things happen. Read the literature. That's sort of obvious, but I think the caveat to that is I found I subscribe to science and try and read it every month. I try and read other things tangential to what I do. And sometimes that inspires me to think about new things or to I try and read articles that aren't just in my field. And that can give me some new ideas or some new thoughts on how other people do things or other fields. And periodically that has led to funding and opportunities that I wouldn't have had if I just focused on my own field all the time. We were talking about before the recording started, the importance of getting to know senior colleagues in your department or in your space or in your company or in your immediate environment. They have a lot of stories and a lot of useful information and maybe help you from reinventing the wheel or learning from past histories. And then just finally, basic kindergarten stuff here, right? I mean, everyone, everyone has setbacks. Everyone gets their papers rejected. Everyone gets their grant reviews rejected by, a bunch of people who have no idea what I was talking about. You got to have a short memory and just keep plugging along. It's like the goldfish thing. You just got to forget it and keep going. Be a good person. I think this is also something that's just not discussed enough. How important it is in this discipline to be honest and be kind to each other and be generous with your time and your ideas. And so many people have done that for me, and I always try and do that for people. And I wasn't in this business to get rich or be famous or whatever. I love science, and when you find like-minded people, then sharing ideas and sharing things and being honest with each other just makes it so much more fun and so much more enjoyable, instead of people being hyper protective and insecure about what they're doing. Good things happen to good people. And then, celebrate it when it works out. I mean, it's really nice. I think sometimes we forget how great this job is and how great, this career is and how great this field is and all these things. And so being able to really enjoy it as it's happening. And, it's very easy to get caught up in day-to-day dramas, but taking a step back and really appreciating, what a great time it is to be in this space and what a great, how great the community is and how great the colleagues are and all the potential for the future. It's really, really exciting. So I think I probably overwent my time there, but that's all I wanted to say. And welcome, Jorge. I will watch the YouTube video later, but congratulations on your successes so far and your career.
Yeah, thanks for the advice as well.
So wonderful talk, Scott. I completely agree with you. Every single point you just made last slide. The best collaboration, I believe, over the coffee or beer. That is what happened to me. My 10 years, longest the collaborator actually over the coffee. And then you don't need to travel. Just your colleague, just knock the door, coffee, and brainstorming, or even talking about random thing. I mean, just that is kind of best collaboration that happen. So I absolutely, agree with you. And then also you just also mentioned, be kind. I mean, now we have the global problem. I wanted to solve more than 10 problem, but somebody scooped me. Now I check my box. I don't need to worry about that problem anymore. So I only focus on 9 more problem. If you scoop me again, 8. So I'm happy, delighted nowadays. So that's why I'm sharing more, with my vision and perspective while I'm, giving a talk. So absolutely, wonderful talk. Thank you so much, Scott. And you are true leaders, and then caring about, the junior faculty and young people. Thank you so much.
All right. Thanks. Good luck, everyone.
Yep. Thank you. All right. Now the main speaker of today with a longer introduction, Dr. Jorge Marchand is an assistant professor of chemical engineering at University of Washington in Seattle and another Washington University. Originally, he is from Puerto Rico. He obtained his PhD at the University of California, Berkeley in the Michelle Chang lab at the Department of Chemistry and Department of Chemical and Biomolecular Engineering. He was a postdoctoral research fellow with Dr. George Church at Harvard Medical School and then Department of Genetics. He also mentioned his name pronunciation should be Jorge, not the George, because George is his advisor. In winter of 2022, he studied as an assistant professor, University of Washington. So congrats again. His group invests time thinking about life beyond the standard building blocks. In particular, the group is developing tools and engineering bacterial hosts that will be compatible with non-standard nucleic acid. This nucleic acid can base pair just like the canonical ATGC base pairs, but are orthogonal in their hydrogen bonding. In venturing into uncharted genetic space, his group asked questions like, what could life do with an ATGC XY codon table? What type of aptamers or XNA gymes could we evolve if we had a much larger selection of building blocks? How do we reinvent modern molecular biology to be compatible with the XYWJ genetic polymers? These are very, very exciting questions, I believe. And I also want to know the answers to these questions. So I strongly believe, he will answer those questions today, also in the future, and contributing to the field. So Jorge, thanks so much for your time today, and please take it away. It's all yours now. Thank you.
Yeah, thanks, Tae-sok, for the very long introduction. I forget if I gave you that info for my bio, if you just came up with it yourself. So as Tae-sok said, my name is Jorge Marchand, and I'm at the University of Washington in the Department of Chemical Engineering And I also have an adjunct appointment with the Department of Chemistry. Now, I have an exciting story to tell you today about the work my group has been doing for the past year and a half or so since we started. It's currently unpublished work, but hopefully it will be out later this year. But I wanted to begin not with my own work, but with a short story about myself and about life and about nature. So this is where I'm from, as seen from space. This is Puerto Rico. It's a small island in the Caribbean, so 100 miles by 30 miles. And I grew up over here in San Juan, and I spent most of my time in San Juan and the surrounding metropolitan area. And something I took for granted growing up was really going to the beaches. I pretty much never wanted to go to the beach, even though we have some of the most beautiful beaches in the world. One of the most famous beaches in Puerto Rico is off the coast of Puerto Rico in a tiny island. It's an island even smaller in Puerto Rico called Culebra. And here you're going to find one of the top 10 beaches in the world. And this is a picture of that beach. So this is Flamenco Beach in Culebra. And now that I'm older, whenever I go back to Puerto Rico, I think I spend about 30% of my time visiting family, and maybe the 70% of my time just going to the beaches. And I generally just sit there in the sand, either thinking or not thinking, honestly, just relaxing. And although this view, sitting from the sand into the ocean, can seem very peaceful, what we're actually observing is this ongoing battle in our oceans, a battle between the microbial inhabitants of the oceans and then the bacteriophages that are trying to take over. Just a few years ago, scientists from around the world made this discovery from one of these marine bacteriophages that should really force us to rethink the possibilities of life. They found that the genome of this phage didn't use A, T, G, or C for its DNA like all of life on Earth. Rather, it used a new set of alphabet. So it used Z, T, G, and C. So in the genome of this phage, the Z, which is shown on the top left over here, let me see if I can get my cursor, yep. The Z base pairs with a T through these 3 hydrogen bonds. And notably, this virus doesn't have any A in its genome. So there's a small change that's going on here, right? We're going from an AT base pair that has 2 hydrogen bonds to this ZT base pair that has 3. But this really small change gives the virus a superpower. The genome of this phage adopts a completely different structure than what you might typically observe in nature. You see, the ZTGC DNA adopts a zDNA form helix, which I'm showing here on the left. And this is a left-handed turn helix. The ATGC DNA, which we've studied for the past almost a full century, usually adopts a B form right-handed DNA helix at neutral pH in water. So what this does for the phage is that it gives it a superpower to avoid a lot of the self-defense mechanisms that the bacteria host has. So for example, this phage is resistant to various restriction endonucleases that would otherwise completely chop up the phage genome. Now, as synthetic biologists, our group takes inspiration from events like this in nature. We like to think and ask, what could we do if we had access to different letters of an alphabet or an expanded alphabet? Now, the work you're going to see today was led by my graduate student, Hinako Kawabe, and involved a great team of collaborators both at the University of Washington and at the Foundation for Applied Molecular Evolution. Now, at the UW, we had Jens Gunlock's group and Andrew Laszlo's group in the Department of Physics, and at FAME, we had Steve Benner and Tuichi Hoshika's group. So I want to introduce you to our world and really put you in our mindset. While you've lived your whole entire life and existed on this planet, really in life that's been dominated by a 4-letter DNA code, so A, T, G, and C, our group thinks about what we could do if we could expand the language of life. Now, In particular, we work extensively with non-standard unnatural base pairs that are orthogonal to the standard genetic code. So I'm showing these on the right. Now, with these additional letters, we can form 6, 8, 10, or 12 letter alphabets instead of just a 4-letter alphabet. So I'm going to be showing you these bases throughout the talk, mostly just their abbreviations. And you don't really need to pay that much attention to their structure. Just know that they are orthogonal in how they base pair, and that the base pairs are B and S, which I'm showing in the top left here, P and Z, X and K, and J and V. Now, for 2 of these base pairs, we have 2 different isoforms. So either SN, which is an N nucleoside, so it's attached to the sugar on this nitrogen, or SC, which is attached to the sugar on the carbon. So we spend most of our time either doing experiments, but also just thinking about how biotechnology can change or will change if it's given access to a much larger DNA alphabet. Relevant to this audience, we like to think, what could we as synthetic biologists do with a xenogenetic alphabet? One that's maybe composed of 6, 10, 12 letters. I'll give you some of the things that we thought about. We could build more stable, more active, or more potent mRNA vaccines. We could evolve novel catalysts that can access new chemical space. So imagine DNA that has the same catalytic capability as a protein. We could expand the genetic code, right, and increase the size of the codon table from 64 codons with 4 letters to 216 codons if we had 6 letters. We could make organisms that are securely biocontained and are never allowed to exchange information with their environment. We could evolve aptamers. So aptamers are DNA that have binding properties like antibodies, but they could potentially be more potent than antibodies, thousands of times more potent, 1,000 times more soluble, and most importantly, thousands of times more cheaper to manufacture. We can also make complex nucleic acid programs for self-assembly, like DNA origami, that has lower symmetry because we can access more complex structure, and even program complex circuits. Now, even though all of this might seem possible today, the reality is that xenogenetics is a technology that's still stuck in the past. It's in this area or the space that existed before the Human Genome Project era. There's no service bioeconomy built around these bases. It's really the Wild West, and you're on your own for all the analytics. So notably, next generation sequencing services don't exist for non-standard bases. Companies that would do Sanger sequencing for you are unable to sequence these bases because they don't have the fluorescent building blocks or the polymerases that will accept these bases. Likewise, you'll find that phosphoramidite synthesis of oligos with XNAs is way too expensive for you to use in routine synthetic biology. Just to give you an example of how expensive we're talking about hundreds of dollars for every XNA that you want to incorporate in an oligo, compared to pennies that it would cost you to incorporate A, T, G, or C. So today I'm going to tell you about how we look to overcome some of these obstacles with a story that addresses these 2 problems. So writing and reading XNAs. And as you'll see, these problems are tightly interrelated. So the solutions in writing are going to give us the ability to come up with solutions for reading. Now, the story that I'll start with first is going to be about writing. Specifically how we developed a simple, flexible, enzyme-assisted strategy for incorporating these XNA base pairs into DNA sequences. Now, the story doesn't begin today, but it actually starts maybe 40 years or so ago, in the late 1980s. Now, scientists back then were studying DNA polymerases, and they found that some DNA polymerases have this non-templated polymerization activity. Specifically, they found that DNA polymerase 1 from E. coli will add an adenosine to the overhang of a blunt end of DNA. If you're familiar with TA cloning, this is generally how you add the A overhangs to your insert. So this is something that we very quickly discovered as a scientific community and very quickly took advantage of. But we thought that this could be a really great reaction for us to make, incorporate XNAs into DNA. So the reaction is completely enzymatic, which is great because we don't have complex organic synthesis capabilities. The building blocks used for this reaction are just triphosphates, which are more easily accessible for XNAs than the phosphoramidites or any custom-built modified building block. Importantly, there's no protecting group required because this reaction should only add one building block. So the question we asked is, could DNA polymerases do this reaction with XNAs? So could we use it to add one XNA to the end of blunt end DNA? So what I'm going to be showing you here is our goal reaction. And what we're doing is that we're using these DNA hairpins that we're using because they only have a single 3' end, which is where the new building block should be added. And the first thing that we did is that we designed an assay to screen for polymerases that can accept all of our XNAs. And the way that we designed this assay is that we made some DNA hairpins that had this non-hydrolyzable 3' end. So in a single pot, we can add a polymerase, a 3' to 5' exonuclease, and a triphosphate substrate. Now, what ends up happening here is that the polymerase is going to come in and add a base to the end, and then the exonuclease will get rid of it, and then the polymerase comes back and adds a base, and then the exonuclease gets rid of it. And this kind of repeats non-productively, resulting in this net reaction where the triphosphate of our XNA is being converted to a monophosphate of the XNA. Now, this monophosphate is something that we can easily detect with LC-MS. So here I'm showing you one example for the Z base of our XNAs. And I'll highlight that this Z base is different than the one from the virus I showed you earlier. This is a synthetic base made by chemists. It just coincidentally has the same abbreviation. So what I'm showing you here is a chromatogram of many different conditions where on the x-axis is time, y-axis is the abundance of the monophosphate for Z. And what you see is that we only see production of the Z monophosphate if we have a polymerase, an exonuclease, and DNA. If you leave anything out, we are not seeing this reaction occur. So this tells us very quickly that this polymerase is able to form, is able to add the building block to the end of DNA. After screening around 10 or so different polymerases, we actually found some that work very well with all the bases that we're interested in. So it works with all the standard bases and all 9 XNAs that our group wants to study. So this was great. But you might be wondering, how precise is this reaction, right? Is it going to keep adding multiple XNAs? Does it give a distribution of products? Is it precise, right? So after we did some optimization, we designed a new assay specifically designed to answer this question about how many XNAs are being added to the end of the blunt end. Now, what we're doing here is that we're doing this tailing reaction, and then we're digesting the product so that we can look at it directly on a mass spec. And I'm showing you here an example again for the Z base. And as far as we can tell, for all the XNAs that we looked at, this reaction is actually incredibly precise. So what we see is that we consume all of the blunt end DNA, and it all gets converted to DNA that has a Z at the end. We don't see any DNA that has more than one Z. We don't see any loss of the building blocks at the end. Now, I showed you here just the example for the Z base, but we showed that this is true for all the XNAs that we looked at. All right, so this seems kind of useless. You're just adding one base. How do we get a base pair? Well, very simply, all we have to do is generate a complementary overhang on another piece of DNA, as I'm showing you here. And then we ligate them together just as you would using like a TA cloning method. And we screened a few different DNA ligases and also optimized this XNA ligation reaction. I'll tell you a little bit about how this reaction proceeds. So unlike XNA tailing, which we saw went to completion, the yield of this reaction was somewhere between 7 and 70% of our starting material. And it's highly dependent on the sequence at the end of the DNA and also the base that you're trying to ligate together. So the actual base pairs that you're ligating. Now, just to give you the most extreme example, which happens with our two different isoforms of S, even though SN and SC both can base pair to B, the yield of the ligation for the SN-B overhang was 70%, whereas the SC was a lot less productive. Now, that said, even though the yield is low, we can get good purity from both of those reactions, which allowed us to develop XNA sequencing solutions. So I just told you how we came up with this way to write with XNAs. And I'll note that this reaction seems very simple, right? But it's actually very deceptively simple because it's very, very flexible, as I'll show you, and very powerful. So I'm going to show you how we use this very simple reaction to develop NGS strategies for XNA sequencing. There's NGS here is next generation sequencing. Now, although there's many different types of next generation sequencing platforms available, we thought that nanopore sequencing would likely be the best possible solution for XNA sequencing. Now, in nanopore sequencing, a voltage is applied and DNA is threaded through a pore, which generates a current that we measure. So the output of nanopore sequencing is just a signal current over time. And it tells us something about the bases that we're passing through the pore. Now, what we really like about nanopore sequencing is that it accepts native building blocks, so you don't need to synthesize anything special. The device is cheap. It only costs $1,000. The runs are very affordable. So the cheapest flow cells are $80, which makes it useful for routine reactions. And because this is the third generation sequencing technology, it's multiplexable, real-time, high throughput, and the measurements are made at the single molecule level. But there's a very big catch. Decoding the signal, so going from current to sequence, requires a model that just doesn't exist for nanopore sequencing. And I'll show you an example of what I mean here. So we sequence a strand of DNA that contains a BS base pair. So I'm showing that right here on the top right. And then we try to base call it using the standard nanopore models. And what ended up happening is that 96% of the reads, the B was being called as an A. And in 82% of the reads, the S was being called as an A. There's nothing intuitive about this. So B is a purine, A is a purine. Okay, maybe they look similar, but S is a pyrimidine, right? The S structure I have down here on the right. And it's still being called as this purine. But maybe this is also not unexpected. Nanopore doesn't know that B and S exists. In fact, it doesn't know that any XNAs exist. It can only classify something as A, T, G, or C. So it's basically forced to make a decision when the right decision doesn't exist. That said, if we were to look just at the raw data, and here I'm showing you an example of two sequences, one that has a Z and one that has a C. So in red is the signal for XNA, in black the signal for DNA. If we were to look at the raw data, we actually do see pretty clearly that there's deviations in the region where the XNA is incorporated. So we see that the XNA signal just shifts a little bit. And in fact, this is significant enough shift that by eye, I could tell that this sequence in red was different than the sequence in black. So what we set out to do was build our own model that can decode this current sequence or current signal appropriately. We should be able to teach nanopore how to read these XNAs. Now, to do this, what we need to do is build our own models for base calling from the ground up. And this is where the XNA tailing and XNA ligation provided us a breakthrough. So we used these two reactions to build these very large, diverse libraries of DNA that contained XNA base pairs embedded in a full factorial standard base pair context. Now, collectively, this means that for every single XNA that we're working with, so we have 9 XNAs, we have around 4,000 unique sequences where the surrounding bases are just A, T, G, or C. In total, this means we have around 36,000 different sequences that we're trying to feed to our nanopore and record the signal. So we made these very large libraries, and then we fed them to our nanopore. And what we end up observing is that for every single sequence, or for every single subsequence, we observe the distribution of signal that's produced. And this is what we're actually using to build our current nanopore base calling models. Now, I'm showing you here in the bottom in orange is just the distribution of signals that we get for this sequence of APAT. What this means that this is the signal produced by P when the base before it is an A and the 2 bases after it are an A and then a T. Now, you see, in nanopore sequencing, the context of the base going through the pore affects the signal that we observe. And just to show you just kind of an extreme overview of this, this is the distribution of all the signals for the base P that we observe when it's surrounded by many different standard bases. So you can see, depending on what comes before the P and what comes after, we have a very diverse sequence, signal sequence produced. So we can take all of these distributions and condense them down into our model. So what I'm showing you here is our nanopore sequencing model, but visualized, where the color is just a heat map for the normalized current signal. And so from the 36,000 total sequences that we started, we can describe this as 2,300 subsequences or 2,300 distributions of signals that we can then build up to describe any model. So we can take these measurements from these distribution and parameterize our sequencing model. So what we care about generally is something like the mean or the median and maybe the standard deviation, and use that to build up any sequence by just sliding each of these subsequences by 1. Now, what's very powerful about this is now we have a way to predict the signal that's produced if our sequence has an XNA base pair. Now, here I'm showing you 2 examples of sequences that we sequence that either have a B on the left or an S on the right. Now, in orange is the real observed signals for the sequence. And in red is the model prediction that we built. In gray, you'll see the standard DNA model, which doesn't know that these XNAs exist. And you can see that the DNA model will usually get things wrong because it's not finely tuned for the different bases that we're working with. Beyond prediction, right? So beyond saying this is what the signal should look like, we should also be able to base call raw data and assign it a sequence. So base calling is the process of going from a raw current and then making the prediction of what that current best fits. So we took this model that has all these measurements and we packaged it into a base calling pipeline. Now, what you're going to be able to do as a synthetic biologist in the community is use our pipeline to go straight from the raw signal to output processed base call data that tells you where your XNAs are. Now, we hope to make it available later this year once this work leaves peer review, but it comes pre-built with all the models for the 9 XNAs that we measured. You might be wondering, how good is this model at all? Like, how good are we at base calling? So here I'm showing you an example of for the Z base. And this is a heat map of our accuracy of base calling for every single possible sequence of the form where the first 3 bases are any standard base, and then there's a Z in the middle, and then the next 3 bases are any standard base. So this is every single possible combination. This is the 4,000 or so sequences that I was talking about previously. Now, in the heat map, red represents high accuracy. So that means that we usually get it right and blue is low accuracy. It means we usually get it wrong, or it's very hard to know what's right and what's wrong. Now, what you can see is that there is a sequence dependence. So there's certain sequences that if there's a Z there, it's actually very easy to distinguish from the standard nucleotides. For example, sequences that have a GAZ or an AAZ, so these lines here that are darker in red, those produce a signal current that's just more unique. And we get these right base calling greater than 95% of the time. Now, a better way to summarize how good your base caller is by generating these response operator characteristic curves. So here, what you do is that you plot the false positive rate, or also known as the false discovery rate, or FDR, against the recall rate, which is also known as the true positive rate. Now, by changing a free parameter, such as the likelihood threshold in your model, you can make this exchange. You can increase your ability to detect an XNA in exchange for a higher false positive rate. Now, this plots the whole entire space, so how you can actually make this type of trade-off. Now, what's important to know about these plots is that the area under the curve is correlated to accuracy. So the actual area for all the bases is just shown here, where an area of 1 is just a perfect base caller. And the gray dashed line represents a completely random guess. So where we are in terms of how good this actually is compared to standard nanopore sequencing is that we are somewhere around where early nanopore base calling models were for ATG and C. So we are maybe where nanopore was 5 to 8 years ago for standard base calling. Now, I'm going to tell you a very quick story to kind of wrap this up. And it's really about how we push this to its limit. So everything that we've done so far has been done in a very modular way, where we're building models for XNA sequencing, but really they are all independently a bunch of 5 nucleotide sequencing models. But you can just apply them however you wish. Now, after making these models, we asked, would it be possible to sequence DNA that contains all 12 letters? Now, something I will mention is that 12-letter DNA that contains all of these orthogonal base pairs had previously been impossible to make through chemical synthesis because of issues with the phosphoramidite coupling steps. So the XNA ligation gave us this way that we can build a new molecule that's never been made before. I'll tell you how we did this. It was actually quite simple. We independently use XNA ligation and XNA tailing to make 4 different hairpins that contain these other these XNA base pairs, so BS, PZ, XK, and JB. Now, we designed each of these hairpins to have Golden Gate sites such that if we mix them all together and do a Golden Gate ligation, they should self-assemble into our 12-letter DNA product. And we saw that even though there's a lot of reactions going on here, so there's 4 XNA ligation and XNA tailing reactions that then had to proceed to a Golden Gate ligation reaction, the 12-letter DNA was still our major product, which is great because we can then throw this into nanopore and try and sequence it. So we then took this and sequenced it to see if we can actually base call it. And despite the overwhelming complexity of a 12-letter model, so you can imagine that with 12 letters, there's 2,000 possible combinations of subsequences for every single signal that you observe, we actually still can decode the DNA appropriately. Now, I will highlight that this is not true for every possible sequence of XNAs. We purposely chose things that we know will be possible to decode because we have a prediction for sequences that are going to be very far apart in signal space. So where is this all going? Can we do better than the first generation nanopore models? The answer is yes. So what we're doing now is following the footsteps that Oxford Nanopore had been taking throughout the years to make their nanopore base callers just higher and higher in accuracy. So what happened a few years ago is that nanopore switched from using these purely statistical ground-up models to machine learning-based models, specifically neural network base callers. And what we're doing now is using long short-term memory neural networks to train models on these very large data sets. We've already seen a very dramatic increase in performance. And just to give you an example, for P and Z, which were our best performing bases before, our accuracy is now greater than 99% for decoding it. So it's orders of magnitude improvement over our current models. Now, the big caveat here is that for the machine learning, we need way more data than we've currently collected and need strategies for scaling up how we do this data collection. So that's all I wanted to tell you about today. If you have questions about XNA, feel free to shoot me an email. I'm always happy to chat. I'm also happy to take questions now too.
Fantastic talk. I love it. Now I learn more about this very interesting topic. I always, wanted to know. Let me ask question first. This is more, this is kind of question I'm asking all the time. I love movies. One of my favorite movies is Matrix. And you probably remember. So Matrix, they start, again. So my question is, if we start universe again and go back, do you think we'll have the same 4-letter word? I'm talking about ATGC, or we may have the 6-letter word or something else.
Yeah, Tae-sung, this is a great question and something that our group also always thinks about. I think in working with these bases, I'll tell you something about them that is actually a little bit interesting. Now I'm going to slide back here to see this B base was actually available to life before life evolved. So it's a prebiotic base. We observe it in the universe, just like we observe guanine in the universe. So it could be possible that life could have used this base to evolve, but it didn't. So, I think the origins of life community will always be torn about whether everything is inevitable and whether or not life will always evolve from the building blocks found in the universe to use ATG and C, or if other possibilities are present. I like to think that maybe somewhere in the universe, there's organisms out there that have 6-letter codes, 8-letter codes. They're just left to be discovered. We found only recently that there's an organism that uses Z instead of A. Granted, it's a virus and not a complex host.
So in that case, another question is, so 6-letter word might be possible, but 4-letter word, but not ATGC, but other combination also might be possible, right?
Yeah. So there's work from our collaborators, Stephen Benner's group and Chuichi Hoshika's group at the Foundation for Applied Molecular Evolution, where they're working with exactly that. They are thinking about DNA that has 4 letters, but the 4 letters are now BS and PZ. Interestingly, it's called alien DNA, but you should definitely check out some of their work on this.
I see. That's very interesting. So I know, some of the paper, earlier, published. I just got the questions. So I want to just ask this question. So Yuju asked question, just curious, do you know what's the selection pressure in nature that makes such X and A base pairs so rare compared to ATGC?
Yeah, so there's a few different things. So for the other XNAs that we work with, they are completely synthetic. B is the only base pair, or the only base, that is prebiotic. The other ones are not present. So B never had, I would say, a great chance at evolving. Whereas A, T, G, C, they're all prebiotic as well. So you have kind of this natural tendency to form. You know, is there, you know, there has to be, I think, necessarily many types of pressure that emerge at the systems level. So maybe the metabolic burden of having a code that uses 6 letters is maybe way too much, right? And not necessary for life to evolve. And then given that, maybe life necessarily evolved to use the 4 letters that were prebiotically present, and the barrier to transition away from this was way too high.
Yeah, this is a very deep question I always ask. So I'm very fascinated by your talk and your answers. So another question from audience is, writing currently looks like a bespoke process. What advances do you think are going to come next? And another question, is it going to be scaled up of this method, a different form of enzymatic synthesis, TDT, or chemical synthesis like current method, but adaptive for XN?
Yeah, no, this is a great question. This is definitely not the method that should scale up, right? We developed this method because it is the cheapest way that a group without phosphoramidite synthesis capabilities, without a DNA printer, will ever be able to make these bases. Ideally, and I tell this to DNA synthesis companies, if my work is successful in creating a demand for these building blocks, they will very quickly follow, right? So how do we scale up? I think the easiest way would be phosphoramidite synthesis. Currently, IDT will make you BS base pairs, but it's $100 to $400 per base pair. So it's kind of not really worth it when I can make these base pairs for pennies. Enzymatic synthesis strategies, I think, are like TDT, as you mentioned, are great. You know, the reason we didn't go for TDT is because you need some sort of chemical protecting group to actually cycle your single addition, which our group, not having those organic synthesis capabilities, can't access, right? And so hopefully, you know, companies will eventually just add these bases to their pipeline. But, you know, there's not enough research. I like to think there's not enough research in this area because, as you might have seen, the tools for working in this area are pretty antiquated. Specifically, the sequencing aspect. I think almost everyone today is trained on next generation sequencing in their design, build, test, learn cycles. Now, without that capability, you really will struggle to do that much with XNAs. And nobody wants to spend like 2 or 3 weeks trying to sequence something that shouldn't take you more than a day.
I see. So, but that's challenging. Because of challenging, you may be more excited, I guess. So yeah. So let me check one more time. So we don't have more question. Now I see the 11:06 of the Central Time. Oh, we have another question.
Yeah. So is the produced XNAs PCR amenable? Some of them are. And there are limitations. Even if they are, there are limitations. So out of the like 4 additional base pairs, we've successfully done PCR with BS and PNZ. This was work shown by collaborators, so Stephen Benner and Chuichi Hoshika.
I see.
For XK and JV, we haven't shown that polymerases will accept these bases. There might be polymerases out there that will do PCR with them. That said, one of the biggest limitations for PCR is that the replication fidelity is not 99.99% like it is with standard DNA. So for B and S, the fidelity is around 90%, which means that 1 in 10 replication events, your B or your S will base pair with the wrong thing, which is horrible when you're trying to exponentially amplify something because you're exponentially losing those X and A's. For P and Z, it's actually a lot better. It's around 99%, which is 1 in 100. But I will also note that that's still not great. When you compare it, if your X and A's were, if standard DNA was only 99% fidelity, that would be considered a very, very, very high error-prone polymerase. So it would be making too many mistakes for a lot of things. But we're getting there. There are groups that are evolving better polymerases to hopefully plug all this into our pipelines. Yeah.
Sure, I believe you. So I want to see more. So in the future, so looking forward to your paper coming out. And until then, we will not release the video. So hope we could release the video soon. So let me close because now I'm seeing 9 after actually the midnight my time. So let me close. So thank all for joining and staying today. We'll meet again on June 29, Thursday, the same time, the same Zoom link, after 3 weeks of break due to 2 conferences I will be attending and chairing in Asia, including metabolic engineering this coming Sunday, and 3 invited seminar I need to give in Asia. We'll have Professor Eriko Takano from Manchester Institute of Biotechnology, University of Manchester, and Professor Dong-Soo Yang from Korea University. Interestingly, both speakers are Asian, and I visited their university last year to give seminars. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speaker. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. So thanks all, and I stop recording. Just give me 1 second.